Week 1 is an overview of the human genome. The learning objectives for Week 1 are to discuss the goals and outcomes of the Human Genome Project, to describe the structure of the human genome, to describe the structure and function of DNA and RNA, and to discuss epigenetics. Genetics is a special branch of biology dealing with heredity and the hereditary material, the gene. It has been defined as the science of differences or the science of variation. The application of genetics to human differences is called medical genetics. In medical practice, the chief significance of genetics is its role in the etiology of a large number of defects and diseases. There are actually several methods for accurately delineating a genetic disease, and these include clinical evaluation of the patient, a family history, which is extremely important, pedigree analysis, and biochemical and molecular measurements. The Human Genome Project began in 1990, and it was an international effort, and it was coordinated by the National Institutes of Health and the U.S. Department of Energy. It was put together to determine the sequence of the human genome and to identify the genes that it contains, to sequence the genomes of several other organisms that are important to medical research. So the human was not the only organism studied. We also looked at the mouse and the fruit fly. To determine new tools to obtain and analyze the data and to make this information widely available. And it was also done to explore the consequences that advances in genetics might have on individuals and society through its ethical, legal, and social implications. Program ELSI. This is extremely important, and next week, when we do the discussion, you will be looking at the history of eugenics. You will be looking at what are some of the things that have happened in the United States and what we need to do to protect for anything happening again. The completion of the Human Genome Project was in 2003, which was faster than we thought was possible. And it was found at that time that the publicly funded Human Genome Project and the private industry funded ones, one by Craig Vintner at the Celera Genomics, had completed the sequencing of the gene-containing portion of the genome. Going back to the ELSI program, as I stated, it was an integral part of the project. And its mission itself was to identify and address issues raised by genomic research. And these issues could affect individuals, families, and society. And in doing this, they focused on the possible consequences in the four main areas: privacy and fairness in the use of genetic information, which includes the potential for genetic discrimination in employment and insurance, the integration of new genetic technologies such as genetic testing into the practice of clinical medicine, the ethical issues surrounding the design and conduct of genetic research with people, including the process of informed consent, the education of healthcare professionals, policymakers, students, and the public about genetics and the complex issues that result from genomic research. Let's look for a moment at informed consent. What is informed consent? Well, before a person has a genetic test, it is important that he or she understands the testing procedure, the benefits and limitations of the test, and the possible consequences of the test results. The process of educating a person about the test and obtaining permission to carry out testing is called informed consent. Informed means that the person has enough information to make an educated decision about testing. Consent refers to a person's voluntary agreement to have the test done. In general, informed consent can only be given by adults who are competent to make medical decisions for themselves. For children and others who are unable to make their own medical decisions, such as people with impaired mental status, informed consent can be given by a parent, a guardian, or other person legally responsible for making decisions on that person's behalf. Informed consent for genetic testing is generally obtained by a doctor or genetic counselor during an office visit. The healthcare provider will discuss the test and answer any questions. If a person wishes to have the test, he or she will then usually read, and sign a consent form. You will be hearing a lot about informed consent. You will be looking at the different types of informed consent that are found at different clinical sites that you will be visiting during this program. The website that I listed below here is the NIH U.S. National Library of Medicine, and there is a very nice reference for informed consent at that site. Now, the Human Genome Project now serves as the foundation of efforts to catalog all human genes, to understand human gene structure and regulation, to determine the extent of variation in these genes in different populations, and to uncover how genetic variation contributes to disease. The human genome recognizes two types of genes, those which have a product that's a protein and those whose product is functional RNA. There are believed to be 20 to 25,000 protein coding genes and 20 to 25,000 functional RNA genes. As more genome sequences were acquired, several new biological disciplines arose. Genomics, which studies the structure, function, and evolution of genes and genomes. Proteomics, which identifies the set of proteins present in a cell under a given set of conditions and studies their functions and interactions. And bioinformatics, which was created to develop hardware and software for processing and storing nucleotide and protein data. The field of genomic medicine seeks to apply a large-scale analysis of the human genome and its products, including the control of gene expression, human gene variation, and the interactions between the genes and the environment to medical care itself. Only 1.5% of our DNA encodes protein. About 20,325 protein encoding genes. Then the DNA sequences comprise the exome. Genes known to cause disorders or traits are cataloged in a database, and this online database is called the Online Mendelian Inheritance in Man, the OMIM. It began in the 1960s, and it was put together by Dr. Victor McCusick and his colleagues at Johns Hopkins. When you look at this online database, we see that the catalog can be searched by gene, by phenotype, by gene locus, and many other features. Each entry is unique. It has a 6-digit number, an MIM. For autosomal dominant traits, the number begins with a 1. For recessive traits, it begins with a 2. For X-linked, a 3. For mitochondrial traits, a 5. And specific genes have a number that start with 6. Each gene consists of a sequence of DNA. Genes, as you know, are the units of heredity. And we've discussed the two different types. Humans are 99.9% identical. The difference lies in the regulation. Traits are produced by an interaction between the genes and their environment. Alleles are variants of genes. Changes in DNA sequence that distinguish alleles arise by mutation. Mutation in sperm or egg cells are passed on to the next generation. Mutations may be positive, they may be negative, or they may be neutral. Francis Crick, an English scientist, said in 1953 that a genetic material must carry out two jobs: duplicate itself and control the development of the rest of the cell in a specific way. And here we have a picture of DNA. This slide is an interesting slide because it shows you how we started off on our road to finding the double helix. And we start here with Frederick Miescher, who isolated the nucleon in the white blood cell nuclei. We then looked at Frederick Griffith, who transferred the killing ability between types of bacteria. This was done in 1928, but we really didn't know exactly what was going on until many years later. We look at Oswald Avery, Colin McLeod, and Macklin McCarty, who discovered that DNA transmits the killing ability in bacteria. This was found in the 1940s. And Hershey and Chase determined that the part of a virus that infects and replicates is its nucleic acid, and it's not its protein. This was 1950. We saw that Levine, Shargaff, Wilkins, and Rosen-Franklin discovered DNA components, proportions, and positrons. And James Watson and Francis Crick elucidated the DNA's three-dimensional structure in 1953. And in 2008, James Watson himself had his genome sequenced. In England, we have two scientists who were working at King's College, Rosen-Franklin and Maurice Wilkins. In 1952, they were using a technique called X-ray diffraction. From this process, they were able to deduce that the overall structure of the molecule of DNA from the patterns in which the X-rays were deflected. And they distinguished two forms of DNA: the A form, which is dry and crystalline, and the B form, which is wet and cellular. It took Rosalind Franklin 100 hours to obtain her important Photo 51, and she obtained this from the B form of DNA. This film, Photo 51, is available for you to look at. You will be looking at it as part of your activity for the week. It is a great historic film. She actually was able to reason that the DNA is a helix with symmetrically organized subunits from her work. Rosalind Franklin herself had a conflict with her co-worker, Maurice Wilkins, and in 1953, without asking her permission, Maurice Wilkins gave her Photo 51 to Watson and Crick. And at this time, we see that Watson and Crick were trying to find out what the structure was of DNA, as well as many other scientists in the world. Rosalind Franklin died of ovarian cancer at the age of 37. And if we look at her ethnicity, she was Jewish, of Ashkenazi background. And she was exposed to a lot of radiation, as you can see from her work that she was doing to get her photos. And it's believed that it was her ethnicity and the fact that she was using the radiation that hastened her disease. So she died of ovarian cancer at 37. And if you're interested, you can look at her history. You can look at her pedigree that you can deduce from her history. And you can see that there was a lot of cancer in her family. So Watson and Crick. Watson was an American. Francis Crick was British. Watson was over in England. He was on leave. And they didn't perform any experiments. Both men liked to work with models. And they used the earlier research and inferences from their model building with cardboard cutouts to solve the structure of DNA. They published their findings in Nature in 1953. And the Nobel Prize was awarded in 1962 to Watson, Crick, and to Wilkins. Because Franklin had died before they were awarded the Nobel Prize, her name was excluded because you cannot receive the Nobel Prize unless you are alive to come and get it.